So we'll now introduce the last speaker of our session, which is Michał Bogdan, and he will talk about discreteness, stochasticity, geometry, topology, and mechanical energy. It's more slides of one. So,
hi, good afternoon. It's a pleasure to be here. So thank you to the organizers and especially for the invitation and the opportunity to talk about our research. So it's going to be probably the most experimental talk of this session. So I'm going to largely talk about raw data from one experimental lab. But this data has some sort of physical mathematical structure, and I will talk about this as well. And there will be some equations. Right. So essentially, the topic that I'm working on is an example of a soft granular material. So soft granular materials are physical materials that consist of grains, which are separated by some lubricating layer of a different liquid, and these grains are soft. So, for example, sand would not classify as a soft granular material. Sand is a granular material, but it's not soft. The granules are quite stiff, but there are some materials in which the grains are not so stiff. And one example would be foam. So it's where foam is something that you have in beer or in a bathtub. Essentially, that means that you have bubbles of a gas phase, which are separated by a thin layer of a liquid phase. And the more similar example, the similar example in emulsions, which is we can elaborate by this, but it doesn't matter. But essentially, an emulsion is similar to a foam, but in an emulsion you have a droplet of a liquid phase, again separated, so droplets of the liquid phase separated by a different liquid phase. And tissues are a bit similar to emulsions in the sense that you get some cells which are also separated by some, well, it can be various media, but often it's the extracellular matrix. And in cells, in tissues, the cells play the role of these individual grains. So here we have an example of a small cluster of circulating tumor cells, which are adherent to each other, and they are floating through a glass microcapillary in an vitro lab. And here we have an example of an embryo that's either allows to sort of adhere to a substrate, and it's just gradually spreading on the substrate, or we have a different embryo that's being sucked into a microcapillary. Yeah, and such subgranular materials. Well, one difference between soft materials and continuous soft materials is that there is this internal length scale, right? I mean, in many materials are completely continuous from the upwards of the length scale of the individual atom, and these materials are not, right? There is this length scale of the individual grain, and even if the material itself is like very big compared to the individual grain, the physics of such materials is different. So they have some complex flows, they have some complex response to mechanical stresses. So they are not necessarily, so I mean, many solid materials just are what we call in physics purely elastic, which means that when you apply stress to them, the change of shape is proportional to the stress. And some like simple fluids are viscous on the other hand, which means that the rate of flow is proportional to the stress. But soft granular materials display viscoelasticity, which means that they have some properties that you know the mechanical response is a bit similar to liquids, but a bit similar to solids in other regimes. They also have plasticity, which means that they can irreversibly break after you apply a threshold stress. There are some memory effects, which means that the materials can be after being applied to some stresses, and after these stresses are released, they do not necessarily return to the previous shape. And there are some avalanches, so there's some avalanching behavior observed there as well. And that's only if we have like quasi-infinite subgranular materials, so like very big compared to the individual grain. But we are interested in either in what happens when you go beyond that, when you go to intermediate length scales. So now what happens if you look at the length scale when the whole system is comparable, or just one of them bigger than the individual grain? What happens then? we had good grounds to expect interesting behaviors, and that's what we found. So we actually first designed a system that allows us to build like a paradigmatic subgranular material, and then we can start with the subgranular material. And this subgranular material is a double emulsion, which means that essentially you have these granules of water which are painted with a dye, and they are separated from each other by a lubricating liquid. And I'm not going to go into the technical details unless there are questions about it. But essentially, this thin layer, this lubricating layer, causes them to adhere to each other, but not to merge with each other, essentially. So normally, if you have two droplets of water that come together, they will just merge, not adhere, they don't. So the way you do it microfluidically is first you in narrow junction, you split the water using the lubricating layer into droplets, and then they flow into a wide chamber, and in this wide chamber they form this emulsion, and then there's a third fluid that splits this emulsion into clusters. So the first thing that we got interested in is what is the dynamics of this it's something that is called actually I just noticed that the title got reversed, but it's a flow focusing junction essentially. So this double emulsion is flowing into a narrowing, and there is a third fluid that is sort of pushing it into this narrowing. And then this liquid splits. Yeah, there is in the literature, there are some known examples already of work being done on the subgranular materials in this intermediate length scale. So when the whole system is not much bigger than the individual grain. So there was a different research group which looked at what happens if you force this granular material to flow in an immediate boundary, right? But then this gets narrower and narrower, and you can see that this system would like to form a hexagonal structure, which is sort of this. In this regime, this hexagonal structure would be sort of the optimal. That's how the optimal packing of subspheres in this dense packing regime would. That's what it would do to form a hexagonal structure. But because this is getting narrower, you have they found some really cool recirculation zones, essentially. So essentially here, the droplet that has 6 neighbors is in red, and the 7 neighbors is in red, and the droplet that has 5 neighbors is in blue. And you can see that there is that essentially this happens all the time at the same point. Essentially, there's like a very regular pattern with which the system is dealing with this frustration. With this sort of competition between like the local preference to have this hexagonal structure, and the fact that you cannot really get it locally because of this external barrier. But I mean, our experiments go beyond that in the sense that that our system has this free boundary, right? So there's no rigid external boundary that enforces this perturbation in a very regular way, but the boundary is also allowed to evolve together with the internal rearrangement, and then how this interplays together is what we are interested in. So if you had a simple liquid in this flow focusing experiment, so a simple liquid that flows here and it's forced into this narrowing, it would either have just regular dripping, so either it's split into quite regular droplets with either multinomial or binomial distribution, or you have jetting with so essentially a single jet of this fluid that's just flowing all the time. But essentially, the question will be what happens when this fluid that you're perturbing is granular, right? And it can sort of only split at the advantage between the droplets. Yeah, and we found some very interesting new dynamical modes. And this was found experimentally, and later it was also reproduced numerically by our collaborators experimentally, and they sort of helped us understand more deeply what happens. So either we can have like this classical regular dripping or jetting, like a single jet, but we also have a very interesting intermediate regimes with either dripping but with very irregular cluster sizes, or jetting, but with a jet with a very fluctuating width, and also with some very infrequent breaking of the continuity of this jet. And this depends on essentially the flow ratio between the external fluid that's pushing this internal fluid and this internal fluid. So the external fluid, so essentially QC is the flow rate of this continuous phase, so this external phase, and QD is the flow rate of this dispersed phase, so this internal phase. And you can see that if the external fluid is flowing slowly, you have this, you're moving towards the jetting. If it's flowing faster, you're moving towards the dripping. But this intermediate regime is what's most interesting. Yeah, so we found some. If you look at the minimum width of this stream in the narrowing, we found some very high fluctuations in the regimes. And also, it's especially interesting if you look at the width of this stream at the entrance to the narrowing. There we found some sort of avalanche-like behavior in the sense that in this regime where the continuity of the jet is not broken, oftentimes you get sort of a slow increase of the width of the stream, but then a very rapid collapse. So a very rapid decrease of the width of the stream. So that's yeah. So that's like an avalanche-like behavior that's quite interesting. And we also have in this regime where you do get a breakup of the continuity of the jet, especially when you're in the sort of intermediate regime. So when the external fluid is not flowing very fast, you get sort of a median of about 6 droplets in a cluster. But from time to time, you get very big clusters of like 30 droplets or even 50 droplets. So we get this very long tail. And we have yeah, we investigated why you get this very long tail. So the first question I think to answer here would be why you get from time to time such very big clusters, even though typically you get more narrow clusters. And it appears that what happens is that because of sort of the matching of the velocities of the external phase and the internal phase, this stream of width of one droplet is quite stable because the velocity of the droplets within the stream is quite similar to the velocity of the external fluid. So they are not sort of pulled too fast and forced to break too fast. And from time to time you get this like very you don't get this all the time, but from time to time you get a stream of the width of one droplet, which is quite stable. So then the question would be why it breaks, or what determines that it sometimes breaks and sometimes doesn't break. And we sort of devised this model where sort of you solve for when the velocity of this continuous phase will be matched with the velocity of the external phase, when the internal phase has this width of one droplet, and that sort of predicted what should be the ratio of the flows of the external to internal phase, and that more or less works. But then I think it's uniquely interesting to see why, because that sort of shows what's the relationship between the behavior of a single droplet and the entire system. So when droplets enter this narrowing, essentially the stream at the narrowing gets more narrow, and finally you get two droplets that are next to each other, and then either one of the droplets can overtake the other. Which is what you see here, both experimentally and numerically in the left panel. Or sometimes none of the droplets manages to overtake the other, and then there is a breakup of this chain. And what I think happens, and what simulations also showed as well, is that essentially this external fluid, it has to flow faster around this wider doublet than here, because it has a more narrow space, essentially. So because of continuity of mass, it has to go faster. So it pushes up at this doublet. If this doublet did not fall, like here, and because of this doublet and the subsequent droplet get like pulled, torn from the previous droplet. So that's one example of how in such a granular material you can really get like the stochastic behavior of an individual grain changes the behavior, sort of propagates the behavior of the whole system, and that's why you get this long tail of very big length sometimes. And actually, just going backwards, what we also observed is that you don't get these very long tails if the narrowing is so narrow that two droplets cannot enter simultaneously. Right? So you cannot have the doublet. You can have like a similar median of the droplet classes, but you don't get these very long tails. Yeah, and then we also looked. So actually, there was this other analogy that we observed with we brought behaviors of clusters of circulating tumor cells. So this first this is the reprint of work done by the Fizycznej group, and they looked at what happens when you have a cluster of 6 tumor cells that are approaching microcapillary. So essentially, these clusters are this cluster of circulating tumor cells are taken from a real biological system, but they are forced to flow for easy observation, essentially in a glass microcapillary. And what they observed is that the order in which the droplets, into which the cells approach the narrowing is different than the order in which they go to the narrowing. So, for example, you can see that the droplet, that's cells number 6, which approaches the narrowing as the last. It overtakes droplet 5 over 3, and they speculated that this might be related to differential adhesion. So, like that some cells might adhere to other cells more strongly. But we also we argue that this might be just an example of this essentially deterministic motion in such a granular material. And we also observed in our as the droplets approach the narrowing here, some droplets overtake the others, right? So here they are numbered in the order of approaching the narrowing, but then some droplets overtake the others. So 2 overtakes 1, 6 overtakes 5, and 4 overtakes 3. Yeah, and then we looked at what happens to the cluster after they are formed. That's another layer of that's sort of a different project related to the first one. So we designed a microfluidic system that adheres this double motion into clusters more regularly. So previously, this stochasticity was the exciting thing that we got. And we tried to understand it, but now we try to eliminate stochasticity a bit and to control the sizes of the clusters. And we're able to do it reasonably well by using a different type of a junction, which would just a cross-shaped junction, which adheres the droplet more regularly. And then we look at what happens with this cluster when it goes to cycles of white chamber and the narrowing. Right? In the white chamber, the cluster is allowed to relax, it's allowed to take its preferred shape. But in the narrowing, it's not that good. Right? It's sort of compressed forcibly. So we studied the relaxation of this process. One thing that we observe is that if the cluster is flowing in a constant width, then it sort of forms almost a crystalline structure, even though it's a finite system. So that's very interesting. But it's still, you can still see that it wants to have this hexagonal structure, and there is some competition with the fact that obviously it wants to minimize the external surface as well. So you can see that the shapes of the external droplets are a bit different. But it forms this hexagonal structure when it's flowing in a period of constant width. But when it's going from the narrow channel to the wider channel, then you sort of see a melting of this structure. And then again, after it flows slowly in this wider chamber with a constant width, then again you see sort of a restoration of the hexagonal structure, and then the melting enters the narrowing again. And then after it goes out again of the narrowing, there's a melting and the relaxation after some time in this constant width. So maybe let's look at it again. Yeah, so now melting, now again sort of recrystallization, and then the melting and recrystallization. So yeah, I mean we looked at some sort of quite mathematically, I would say rudimentary measures of what is happening with the system. So if any of you has any suggestions for more stuff to measure, then that's an interesting thing I think to talk about. So one measure that we looked at is just this so we define the parameter G3, which essentially for any droplet you define it by in terms of the angle of its in terms of the angle between like two neighboring neighbors of the droplet I, right? You look at the angle between neighbor J plus I and the droplet I and the neighbor J. And if you look at so essentially it's a perfectly hexagonal structure, this would be 60 degrees, right? And we define the parameter that essentially penalizes this distortion from the 60 degrees. And we actually found that, so on this plot you can see that the white parts are when it goes to a period of a constant width of the channel, and the gray parts are when the width of the channel changes. And then you can see that this parameter decreases when it goes to this rearrangement. But it's not sort of a perfect parameter in the sense that it measures distortions from the hexagonal, from the like perfect algebraic hexagonal structure. But if you have a hexagonal structure that's a bit squeezed in one direction, then it's not going to catch that. So it's sort of so for example, you can see a decrease of this parameter G3, even though there is no rearrangement of the droplets, but only a sort of a plastic squeezing of the whole system. So it's not it doesn't discriminate between certain kinds of processes. So it's pretty rudimentary. Another process that we sort of measured is that's also quite simple is the number of T1 processes at various points of this evolution. So T1 processes is two droplets, if four droplets swap neighbors, right? So A neighbors B and then C neighbors D essentially. So you see when the merging happens, and well, you also can look at the standard deviation of the number of nearest neighbors, so the droplets in the rim and in the interior. So the droplets in the interior should have six neighbors in a perfect hexagonal structure, and the droplets at the rim should have four neighbors, unless they are going at the edges. So if they are at the edges, they should have three. And essentially, for any cluster, in an ideal hexagonal finite cluster, we should have six sort of topological, in the sense that six droplets at the rim should have three neighbors and not four. And here we can see that after a period of relaxation, it's almost like this. So essentially, there's one droplet at the rim that has five neighbors, and then and seven droplets that have three. And in the interior, you have one droplet that has seven neighbors and one that has five. So it's almost like a topological dipole, right? And apart from that, it's a perfect hexagonal structure. But during this period of rearrangement, you can see that still the median is six neighbors for droplet in the rim, and in the interior it's four droplets outside, essentially the distribution is much wider. And now I think that physically it's sort of most interesting for us is that we looked at how elongated the droplets are. So here there's a few films that I will allow to play. Essentially, So essentially, the point is that. Let's see this again. Essentially, the point is that when this cluster is moving, I mean, the rather the droplet, the more elongated it is, and you see that when it starts to move, first you have this wave of contractility in the middle of the cluster, but then it's sort of distributed. It's interesting how it gets distributed. So you can see that it disappears faster in the interior of the cluster than this sort of elongation gets redistributed to the rim. But it's quite subtle to see just based on the colors. But it becomes clear when we plot this. So we define this circularity parameter, which is essentially the area divided by the perimeter square. It's normalized to be one for a perfectly circular droplet. And essentially, the orange data is the data for the droplets in the interior of the cluster, and the blue data plots are data plots for the droplets at the rim. And there are two relaxation units. First, when the cluster goes into this first white chamber, and then when it goes into the second white chamber. And you can see that before going into this chamber, already the droplets in the rim were a bit more elongated. And that might be like a static static effect in the sense that they some like static energy minimization. But you can see that after this period of dynamical changes, this difference is bigger. I mean, all of the drops elongated very rapidly during this melting, but then the droplets in the rim have a problem recovering this shape, and we see that it's probably like a dynamical effect related to the fact that the droplets in the rim they sort of topology. I mean, they have a droplet, they have less opportunities to rearrange in the sense that a droplet in the rim has only 3 pairs of neighbors between which it could go to swap neighbors if it's very stressed, right? I mean, for a droplet in the rim, it has 6 neighbors, so there are ways it will escape. It has to accumulate very much mechanical distortion. So we think that in this way, sort of this, the place where the droplet is located in this cluster, so the topology of this little system really influences this response to mechanical energy. So the physics and energetic properties of this cluster, and there is this effect originating. So I think most of the slides I would like to thank my collaborators. So the head of my group, Dr. Amuzowski, and the very fabulous team here in the UK, I've been working with our collaborators in Italy and in Sweden who have been doing simulations and machine learning to track these droplets. Yeah, thank you.